Have you ever wondered what actually triggers a seizure in epilepsy? To understand that, we first need to know how a single neuron fires an action potential, because seizures begin when this fundamental process goes out of control. Firstly, as the statistician George Box famously said, all models are wrong, but some are useful. In this video, we'll use the metaphor of a race to illustrate this delicate process, but keep in mind that the underlying biology is far more complex, and no metaphor can capture every detail with perfect accuracy. Firstly, like the beginning of a race, right before it starts, the membrane starts out peaceful and calm. At this time, its membrane potential is around -65 millivolts. This is determined by the imbalance between ions inside and outside the cell. Most importantly, this is determined by the ability of the potassium cations to leak slightly out of the cell. However, the sodium channels are completely closed, and the neurons inside is negative relative to the neurons outside. This is called the resting state, where everything is in balance or equilibrium. This is similar to the very beginning of the race, where all of the runners are lined up, but no one has started running. This is because the two forces in balance, the gradient of diffusion and electrochemical attraction between positive and negative charge, is stable. This is further stabilized by the sodium-potassium pump, maintaining the correct concentration of sodium and potassium ions inside and outside the cell. However, once there is a depolarization from this resting potential, -65 millivolts, or when the cell membrane potential goes from negative to positive thanks to large cation influx, which can be caused by any sort of synaptic input, makes the membrane potential get closer to the required threshold for the voltage-gated sodium channels to open. This depolarization can be considered the starting pistol of the race, where it triggers the runners to go. Then, when the sodium cations flood into the cells, following diffusion through the concentration gradient, this causes a huge change in the membrane potential toward the Nernst potential of sodium, which is around 60 millivolts. The Nernst potential of sodium means the membrane voltage at which the electrochemical forces that make sodium enter the cell exactly balances the force that pushes it out, meaning there is no net movement of the sodium ions. Similarly, the Nernst potential of potassium means the membrane voltage where the two forces pushing on potassium ions in and out of the cell are equal. As the membrane potential increases, this represents the rising phase of the action potential. In addition to the rise in E and A, Hodgkin and Huxley, two famous scientists, used a technique called voltage clamp experimentation to find that the massive and rapid influx of these positively charged ions led to a rapid increase in the sodium conductance, or GNA. This became important because as the GNA rose faster, so did the membrane's depolarization, eventually leading to a positive feedback loop until the voltage-gated sodium channels begin to inactivate. However, in a relay race, can the first runner keep running forever? No, they don't have enough stamina. This is similar in the neuron, although it happens at a much smaller time scale. In the first leg of the race in the neuron, which is represented by the sodium cations rushing in, after around 1 millisecond of inactivation, the channels close and lock. This means that these ion channels are completely inactivated, even though the membrane is still depolarized. However, right at the same time, the next runner is there to receive the baton. In the neuron, this next runner is the potassium channel. These channels usually become slowly activated after the neuron becomes depolarized, allowing the potassium ions to leave the cell. Because there is this flow of cations outward while the channels allowing inward influx are closed, the membrane potential can be set closer back to the Nernst potential of potassium, or EK, which is much lower. This return to equilibrium, according to Hodgkin and Huxley, means that as GNA decreases while GK increases, there is a depolarizing slope that represents the downward side of the action potential. Then, after the voltage has reached back to equilibrium, or resting level, the potassium channels remain open for a while after. Because of this, the potential actually briefly undershoots and becomes more negative than -65 millivolts. This is called the after hyperpolarization, or undershoot phase. If we think about it like a relay race, at this time, the baton has been passed to the very last potassium runner who finishes the last lap. Eventually, the potassium channels close and the membrane potential can then return back to resting potential in order to prepare for another action potential. However, when these athletes come back together to compete, they need to have rules to keep them in check. There are two of these in the neuron. The first is the absolute refractory period, which starts when the sodium channels inactivate. During this time, no new action potentials are allowed to happen because none of the sodium channels are available to open. Bringing it back to the race metaphor, it's similar to how after the first runner runs their first lap, they have to recover before continuing to run. After this, which is the absolute refractory period, there is the relative refractory period. During this phase, some of the sodium channels can recover, but many of the potassium channels are actually open, leading to a hyperpolarized or more negative membrane potential. This means that it is possible to activate another potential during this period, but only through a more strong, intense stimulus, which requires extra work and more depolarization. After the GK is normalized and the sodium channels are back at their baselines, the race is finished and the track is ready for the next set of runners. These two scientists I talked about earlier, Hodgkin and Huxley, actually took these periods, this race, and figured out how they could represent it using math. They decided that they wanted to take every single ionic current and add them together to get the total membrane potential. In the equation, though it is quite complicated and relatively difficult to derive, each of the conductances represented in the picture, GNA, GK, and GCl, is a function of time and voltage. Essentially, based on the concentration of these ions, and therefore their relative conductance in each phase of the action potential, this model can accurately predict all the phases of the action potential, giving us a graph of the values of the total membrane current over time. This was a really important discovery and development because it allowed us to understand the actual physical workings behind the neuron and fully understand the physiochemical mechanisms by which it functions. And then, once an action potential starts, it's able to travel, meaning it can pass the baton down an axon, which starts the same action potential in the next neuron and the next. Due to this, the neurons can convey information by the rate of the action potentials occurring per second. And when this amazingly timed sequence breaks down, when the channels open too easily, close too slowly, or inhibition fails, the action potential race can digress into this sort of uncontrolled synchronized firing. This runaway excitation is actually what the physiological basis of a seizure is.